So hello, we've reached the top of the hour, so we're ready to start today's live webinar by Lipid Maps. So today's webinar will be presented by Prof. Steve Blanksby, and the title of Steve's talk will be Empowering Lipid Discovery Through Isomer-Resolved Mass Spectrometry. So I'm Bill Griffiths from Swansea University, and I'm host of today's seminar. So first of all, a few housekeeping notes. So the webinar is being recorded and will be uploaded onto the Lipid Maps website and our YouTube channel within the next 48 hours. The webinar will be around 45 minutes long, allowing 10 minutes for questions at the end. Please use the question and answer function throughout the presentation to ask questions, and we'll unmute you at the end of the webinar to ask your questions directly to Steve. Today's webinar is being sponsored by Avanti, and we're grateful for their ongoing support. So before we start, a few words about Steve, our speaker. So Steve has a PhD in chemistry from the University of Adelaide and has done postdoctoral studies in Europe and the US and the USA. He returned to Australia to take up a position in the University of Wollongong before moving to the Queensland University of Technology in 2014, where he's Professor of Chemistry. Steve's research is focused on advances in mass spectrometry to empower molecular discovery in chemistry and biology. Steve has published more than 200 papers, and he serves as associate editor of the International Journal of Mass Spectrometry. He's currently vice president of the International Mass Spectrometry Foundation. So Steve, it's a pleasure to have you here to talk to us. So the floor is yours for the next 45 minutes.
Thanks very much, Bill. Just confirming you can hear me okay? Thank you. So, yeah, thank you, Bill, for the introduction, and thanks very much to Lauren and Valerie for the invitation to join in the Lipid Maps seminar program. What I wanted to talk to you, I know that the seminar program very much focuses on the biology of lipids. And that's crucially important. But I guess what I'd like to sort of focus on today is the technology that enables us to discover, I guess, the molecules of life and the lipids in particular, and some of, I guess, the contributions that we are making to that, but more to introduce this idea that by sort of advancing the toolbox of lipid mass spectrometry in particular, we can advance our understanding of the lipid biology and indeed discover new lipid metabolism, new lipids, and new biology. So that's the aim today. So I guess perhaps it's motivated by this quote, which I really like, from Freeman Dyson, so a Princeton and Cornell physicist. And in his book, Imagine Worlds, he describes new directions in science are launched by new tools much more often than by new concepts. The effect of a concept-driven revolution, is to explain old things in new ways. And the effect of a tool-driven revolution is to discover new things that have yet to be explained. So, you know, this is kind of what I want to drive at today, that as we keep advancing that toolbox, you know, getting the right tool for the job, we will actually discover genuinely new things that will require new understanding of biology to be able to explain. Of course, the famous Australian scholar, Mick Crocodile Dundee, said this much more succinctly when he said, that's not a knife, this is a knife. So it's about having the right knife for the job or the right tool for the toolbox. So what does our lipid toolbox look like at this point in time? And I'd argue that there's, and you know, I'm happy to have the argument or the discussion at the end. There's really sort of two main toolboxes. One around GC mass spec, which really tells us a lot about the, I guess, the small lipid molecules and metabolites. And so from this, we've learned most of what we know currently about, say, fatty acid metabolism. Conversely, particularly over the last 20 years, with the sort of advent of sort of electrospray and electrospray tandem mass spectrometry and LCMS, we've learned a lot more about complex lipids, membrane lipids, storage lipids, etc. And so this sort of has informed and taught us more about complex lipid metabolism. But I guess what I want to draw your attention to is to sort of what are the strengths and limitations of both of these techniques as they currently stand, I guess, in their conventional form. So if we think about the GCMS toolbox, so this is in a conventional way, taking fatty acids, maybe free fatty acids or saponifying esterified fatty acids, and then typically derivatizing those to fatty acid methyl esters, and then running those fatty acid methyl esters through GC to get, some degree of separation, and then subjecting those to, typically it's some form of chemical ionization, but I guess still more typically is electron ionization. So here's some data from our lab. You don't really need to know too much about the details of this. It was part of just some benchmarking that we were doing. And so what you see in black here is the GC trace for 3 reference standards. And these are all 18-1 fatty acids that have been derivatized as methyl esters, and they differ only in the location of the carbon-carbon bond. So either at the 7 position with respect to the methyl end, the 9 position, or the 10 position. And we're just sort of using methods straight out of the textbook, and there's nothing too fancy here. And I understand there's many people better at GC than we are and can probably do a better job. But I just do it to alert you to a few things about GCMS, right? So in one respect, GC obviously is the gold standard in terms of separation, but you can see here that for the N10 and the N9 isomers, the separation under conventional conditions is incomplete. Okay, and so if one of those species in a native extract, in a biological extract, is lower, it may be questionable whether you would be able to call it out as an individual species. And so in cases like that, with GCMS, what we would conventionally do is go to the mass spec dimension and look at the differences in the mass spec and do extracted ion chromatograms. And here we sort of hit the snafu that the electron ionization mass spectra of all these fatty acids are 1-to-1 and onto, they're identical. So really for the GCMS in its conventional form, we're very much just relying on retention time to assign identity. And this becomes problematic in terms of discovering new fatty acids or new smaller lipids, because we really just don't have a detailed structure elucidation dimension coming from the mass spectrometer, even though the GC is as I say, gold standard in terms of separation. So if I flip now to talk about where we're at with LCMS, we'll be looking at a complex lipid and complex lipid extracts. So in this case, we get less separation, so overall the peak capacity is less. And with complex lipids, they're tending to merge anyway. And so for something like a phosphatidylserine, there will be potentially multiple versions of a phosphatidylserine with 36 carbons and 1 double bond. And I'm sort of just doing a quick data dump from the LIPID MAPS database there. So just the things that are in the database, there's 16 potential distinct molecules there. So in LCMS, what we would tend to do is, again, rely on the mass spectrometry dimension, particularly the tandem mass spec or the collision-induced dissociation dimension. We would pluck out this mass 788 subjected to collision-induced dissociation. We get a beautiful mass spectrum here. That mass spectrum confirms that we are actually, in this case, looking at a phosphatidylserine. And there's lots of beautiful redundant fragmentation that tells us that we've got an 18-carbon saturate and an 18-carbon monounsaturated. So that goes some of the way to identify the structure. So this is kind of the fragment map that I've just shown you. But then there's lots of detail in that structure that we don't get from the mass spectrum. So to put that another way, that mass spectrum allows me to exclude certain isomeric forms. So for example, this acyl chain compositional acyl chain isomer with different chain lengths or double bonds. But there are a whole range of isomers which would vary, for example, in the relative position of acyl chains on the glycerol backbone, the position of carbon-carbon double bonds, or even, you know, various stereochemical or geometrical arrangements. So all of those are sort of, you know, common motifs, and they can't be excluded based on the tandem mass spectrum. And we're not going to resolve them, or, you know, it would be hard work to resolve them based on conventional reverse phase chromatography. So if we sort of come back to, you know, why does this matter? And this matters because we sort of now have this disconnect. We can learn something about the fatty acid metabolism from the GCMS, noting the limitations that I've talked about. And on the other hand, we can learn about sort of a different corner of the metabolism from the LCMS. So we can still, you know, say what's going on with phosphatidylserine as sort of a class or a sum composition level. That tells us a little bit about areas of metabolism. But we can't sort of connect the dots. Which fatty acids are involved with the phosphatidylserines, for example, which ones are involved with the phosphatidylcholines. So, you know, rather than this just being an abstract concept, here's a real life example from Sarah Marie Fent's group, really elegant piece of work where they've really taken, you know, the two toolboxes of lipidomics as they currently stand and deployed that in sort of the cancer lipidomics context. And their discovery, if you like, by paying very careful attention to the GC data, was that indeed, in addition to the oleic acid and the cisphosphenic acid that one would sort of expect to be in sort of a mammalian cell or a mammalian tissue, they also observed what was essentially just a shoulder on a peak. And this is this N10 isomer, so with the double bond 10 positions from the methyl end. And this speaks to this idea that, you know, in terms of canonical metabolism, with the exception of sebocytes, some unusual mammalian cell types, this N10 double bond position shouldn't really be there. So essentially here we're relying on some very careful GC work to discover that in the context of these cell lines and in the context of the tissue, this different metabolism, this non-canonical metabolism was taking place. In parallel, they were able to sort of profile what was happening with the complex lipids, so really elegant work from Johan Swinnen here to identify, you know, across 200 or 300 different molecular species at the sum composition level, what was going on. And what you can see there is that, you know, between friends, there's not a lot of variation in those species, and yet there was variation in these fatty acids. So this is a really strong example of where the disconnect that we have in the technology, in the toolbox of lipidomics, doesn't allow us to connect one bit of metabolism to another bit to see where that's actually connected. And so I guess that's the gap that we've been trying to bridge to see whether we can bring new technology that will help can make that connection and present a fuller picture of the metabolism, but perhaps even discover new molecules and new metabolism. So to do this new work, we are relying on some very old work, in spite of the fact that, you know, it's often said I didn't invent ozone. And in fact, I'm by far and away not the first person to deploy ozone in lipid science. So some beautiful work from the 60s taking, for these GC type workflows, taking lipids offline and undergoing ozonolysis in solution and then doing GC of the products. You can identify then, in essence, the two pieces that would have ultimately been connected. Challenge, of course, is when you get a very complex mixture with lots and lots of lipids, how do you know which two pieces go together? More recently, work from Bob Murphy's group in the 90s sort of with this idea that you could do ozonolysis on a surface And again, it's offline outside the mass spectrometer. You create these ozonides. So this is the mass of the lipid plus 48 mass units for ozone. But then when you take that inside the mass spectrometer, you activate that, and that breaks the ozonide, and you get cleavage at the position of the carbon-carbon double bond, and you can identify it that way. So again, the challenge here is you're really taking a complex mix, what's already a complex lipid mixture that you've got offline, and you're making it more complex by sort of, adding in this chemistry before you put it in the mass spectrometer. So very powerful, but becomes limited to sort of scale up to a really complex lipidome. So I guess our wrinkle on this was to, if you like, stabilize the ozone, bring it inside the mass spectrometer, and use the ability of the mass spectrometer to select out the species of interest. What's the sum composition that we're interested in? We can select that by mass. And later on, I'll show you examples where we can, then deploy all the tricks of LCMS in terms of LC separation. We can do mobility separation and things like that, and then bring the ozonolysis in on the purified or the individual molecule. So in this example, just to show you that this works, if you take this phosphatidylcholine, it has by mass only one double bond. We can mass select that. We did these experiments just on a simple benchtop ion trap mass spectrometer. We bleed in a tiny amount of ozone, and we see a gas phase ion molecule reaction, which produces two ions. And because of the ozonolysis chemistry, as I've described, is so well trodden and well understood, you can write a chemical mechanism that allows you just to predict what the masses or the mass to charge ratio of the products you would see, depending on the position of the double bond. So in many ways, what's nice about this is this knowledge allows us to do de novo discovery. I don't have to have a reference standard for every lipid I'm trying to describe, which is just as well, because there are many, many new lipids for which these standards don't exist. You will have seen that on that previous slide that in those early days, individual scans, not spectra, but individual scans would require a significant amount of time, simply because the amount of ozone, it's a relatively slow chemical reaction, and the amount of ozone that was required that could be delivered was relatively low without sort of interrupting the normal function of the mass spectrometer. So what we've been doing sort of in the intervening period is to try and, accelerate the rate of the reaction. And the simple way to do this is to add more ozone, which means you look at different ways and different geometries of instrumentation that will allow you to deliver that higher number density of ozone without affecting the instrument performance, and ultimately to be copacetic with the duty cycle of the instrument. So just to give you an example, if you squirt some olive oil into these various instruments that we've modified over time, and you pick out the trial tag, you can see after 20 milliseconds, you have to magnify 1,000 times to see the ozonolysis products here on the old ion trap that we used on the benchtop. When we move to triple quadrupole and Q-exactive geometries, so HCD cell type instruments, you can deliver more ozone. And so you can see same molecule under the same conditions. We only need to magnify that signal 50 times, okay, for a 20 millisecond acquisition. So this is becoming highly usable. And in fact, we've used these geometries to look at quite complex lipidomes and draw quite a lot of data and on a meaningful timescale. And then if you move to ion mobility geometry instruments, not necessarily in the data I'm going to show you are we doing ion mobility, but we can. Here we're really hijacking the ion mobility regions because they deliver a high gas pressure, which we can substitute for ozone. And so a lot of the data I'm going to show you today will be taken either on an Orbitrap type instrument in a HCD cell, or they will be taken on a Synapse type instrument where we've delivered ozone into the ion mobility region. And just to pause to give credit where credit's due, so a lot of the instrument work, the modification and so on of both the hardware and software to sort of bring about these data was done by David Marshall and Beric Pode here at QUT. So if we then just deploy this capability against, we're very intrigued by the putative, the discoveries from Sarah Marie Fent's group on particularly these unusual sites of unsaturation, and to see if we could close that gap. So we sort of would know from Sarah's work that there are these unusual double bond positions in the fatty acids. Could we actually see those within some of the complex lipids, some of the carriers of the fatty acids? So we were fortunate to be collaborating with a group here at QUT who were doing a lot of work in and around prostate cancer. So what I'm showing you here is a range of prostate cell lines, a couple of sort of benignish cell lines and some malignant cell lines. And what I like about this slide is these are more or less the raw data. These are OSID mass spectra, all mass selecting out this, this major mammalian phosphatidylcholines. In most mammal membranes, this is the largest or most prominent lipid by some composition. And if we do the ozonolysis on that, you can see for these two cell lines at the top, you see an N7 variant. There's only one double bond, but we also see an N9 variant. So there's immediately at least two isomers present here. But then when we look at this malignant cell line, this PC3 in particular, even in the raw data, we can see this N10 double bond position, meaning that we've actually got 1, 2, 3, minimum 3 isomers for this one sum composition in this cancer cell line. So it was quite a striking result. We were pretty excited that the technology could reveal that even in the raw data. And so this slide gets a bit complex, but I'll sort of walk you through it, because then we tried to look at, kind of the whole data set. So here are the cell lines across the top here. And this is kind of the breakdown along the top, more or less by major lipid class or major membrane lipid classes. And you can see there are some variations there between those. So this is sort of comparable to the data that I showed you earlier, sort of that Johan Swinnen's group were able to acquire. And you do get some separation based on, looking at what the general profile of the lipid subclasses in the membrane are, but the separation statistically is not that great. If you break that down by some composition, and I guess, the top half of this slide sort of represents, I guess, conventional LCMS or LCMS-MS kind of lipidomics. You can see some of the numbers of carbons and degree of unsaturation moving about a bit, cell line to cell line. But again, many of these cell lines at some composition level are still clustering close together. If we look just at the unsaturation profile, just of that one monounsaturated lipid, that PC34-1, we actually see quite stark variation. So that it becomes a real sort of phenotype of these different cells, is just the pattern of unsaturation carried by that major monounsaturated membrane lipid. And you can see that here in the PCA plot, that these things then splay out really nicely in phase space. So in terms of being a phenotypic marker, we're quite excited about using these double bond profiles. And we've done some work with Zhang Yang and Yu Xia at Tsinghua University in Beijing. And what they have, these beautiful little mini mass specs, which is about the size of a sort of a desktop computer, if you like. But one of the genius pieces of it is they run at a relatively high pressure in the mass spec world, relatively high pressure, which means if we dose in a little bit of ozone, the number density of ozone is actually quite high. And so we get very efficient ion molecule reactions. And so what we were able to do is just take these sort of pinpricks of tissue, load those in the nano ESI, squirt those in, and just mass select out the ion of interest and look at its profile of unsaturation. So there wasn't evidence in this particular profile of necessarily aberrant unsaturation or unusual unsaturation, but even just looking at the ratio of the two sort of canonical 18-1 isomers carried by this complex lipid gives you statistically robust sort of phenotyping of that tissue. So on the one hand, this information or the technology will enable phenotyping, and using this isomer resolution is clearly giving a very tight and reproducible phenotype. But moreover, we can start to connect what's going on in these complex lipids to the underlying metabolism. So what you're comparing here in the bottom in panel D there is the expression of more or less the two enzymes that are responsible for both the canonical desaturation in SCD1 and the non-canonical desaturation in FADS2. And there is some correlation there as to what's going on, but it's clearly a little bit more complicated than that. And so that's where, once you've made a discovery of something new, can the technology sort of enable you to demonstrate where this is coming from and how this is happening? So what we elected to do there was to use the same OzID technology in concert with stable isotope chasing experiments. So feeding these prostate cancer cells with carbon-13 labeled palmitate or carbon-13 labeled stearate. And so what you can see is, when you do this, you can then inhibit one or other of these two desaturation enzymes, these two desaturases. So if you inhibit one, you can see the other one sort of ramp up in its activity. So particularly notably here, if you sort of supplement the cells with this TOFA and you feed them this carbon-13 labeled palmitic acid, you really switch on the N10. So the FADS2 almost takes over in terms of doing the heavy lifting for the desaturation. What was really striking, though, is that I guess the canonical idea would be that you desaturate the palmitate and then you elongate. And we could actually see that by the addition of two unlabeled carbons in this study. But I guess what was more surprising is that if you put in the carbon-13 labeled stearate, you can get direct desaturation. And you can see that happening at the N10 position. So this would suggest that the FADS2 is able to operate both at, I guess, the delta 6 position as well as the delta 8 position. And what was even more intriguing is we sort of got these other desaturation pathways switched on. So here we started to see the N12 pathway opening up, and even the N13 pathway. So these would suggest that there's even a greater variety of pathways or variety of substrate enzyme interactions that could be activated within these cells by essentially shutting down SCD1. So if you sort of lay that all out, this complicated figure, but what we tried to do is to say, okay, well, what we can see now based on this technology is that, if we overlay in gray the sort of what's in the textbook more or less about, the desaturation and elongation of fatty acids with what we have evidence for from fatty acids carried by the complex lipids, the cell has the potential, has the machinery to generate this quite significant repertoire of isomeric forms of these lipids. And so this sort of poses really fascinating questions. So, it becomes firstly likely that this is broader than just, SCD1 versus FADS2. There's evidence here from the N13, for example, that there's activity from FADS1 doing this delta 5 desaturation. So again, much broader capability of these cells, I should say, and essentially the mammalian enzyme system to generate a wide range. There's kind of a wide range of capability from this system. So what might that actually mean? Well, I mean, you might say, there's a couple of ways to think about this. The cells could be compensating and just saying, oh, well, N10's nearly as good as N9. And so if I can't make enough N9, I'll just make N10. But I guess there's enough information or biophysics known about these. I find this quite profound. This is a really nice meta-analysis by Anton de Kroon's group. And what they found by sort of delving back into the literature is that shifting the double bond position even by 1 position, so moving from 9 to 10, could shift the phase transition temperature of the lipid by up to 7 degrees. I mean, that's huge. And so if you think about, if you overlay that with what I've just shown you, this plotted in a different way, so these are 3 of the different cell lines. Here's the major membrane lipid of these 3 cell lines, all sort of prostate derived in some way, which, vary from, 0 N10 through to 50% N10. So that 50% might translate to, somewhere between 4 and 8 mole% of the whole membrane. So what you would predict based on this is that the membrane properties of this cell line are profoundly different to those of the other 2 cell lines. So this sort of piqued the question of, just how many of these, these different variants might be there. And this is a little bit of an aside, but it's also about connecting those 2 bits of metabolism, the fatty acid metabolism with the complex lipid metabolism. And what we found, and I don't really have time to drill down into how we do this, but it's a combination of using collision-induced dissociation and ozone-induced dissociation, is that you can actually find that in addition to the variation in the double bond positions, there's also quite a lot of variation in the SN positions, so the positions of substitution on the glycerol backbone. And moreover, there seems to be sort of a strong interesting different associations of different double bond positions from different SN positions within the molecule. So if you take, for example, the N7 variant over here for PC 16.0.16.1, it is predominantly has the 16.1 in the SN2 position, which again is the textbook idea. But when you go to something like a 14.0.18.1 variant, then the unsaturated fatty acid is primarily in the SN1 position, even for the same double bond position. So there's lots of degrees. The cell is making a smorgasbord of these isomeric lipids. And there's got to be, and this is where we're starting to speculate, this has got to be connected to both the lipid metabolism and the function. So where this might be happening in the cell, so it might lead you to hypothesize that indeed different classes of lipid might actually express these isomers differently as a consequence of the local metabolism and I guess the local functional need of the membrane. And so we've got enough evidence for this. We don't have, the whole picture yet. But here's the PC34-1 data that I was showing you just before. And if you compare that to the phosphatidylate and isotyl with the same number of carbons and the same number of double bonds, you get quite different distributions of the same double bond isomers. You do the same thing for the phosphatidylglycerol, you even see, different isomers altogether entering the fray. So this got us wondering about how much, if we sort of do the conventional thing and hydrolyze all the fatty acids, how much does that actually, what we get from, say, the GCMS, even capture the complexity that might be going on in the different lipid subclasses? So it left us with this idea that we might need to take a much deeper dive just into the fatty acid pool by itself. So almost go full circle and say, well, how many fatty acids are there? And so to do that, we've gone back to the extraction and hydrolysis, the conventional first part of the conventional fatty acid analysis. But rather than a GC method, we're looking to exploit the advances we've made with the LC methods. And so instead of derivatizing to fatty acids, we're derivatizing to these fixed charge AMPP derivatives that Michael Gelb's group introduced a few years ago. And so what you get is a very good chromatographic behavior on reverse phase, and you get a really strong ion signal in the electrospray. And so that allows us to just deploy the ozonolysis to pick out the carbon-carbon double bond positions. So when we took that deeper dive, just in the fatty acid pool, and this work was done by a talented postdoc, Phil Menzel, who's recently just left the group, you can see that here, over this side of the LC, these PC3 cell lines, you certainly see the N7 N9 and N10 isomers that we were seeing carried by the phospholipids, but we're also seeing evidence for the N12 and N13. So these things were sort of dropping off the detection limit of the GC analysis because of below the dynamic range, I guess. But also we wouldn't have had reference standards for these to assign the structure by GC, but here we can do this de novo structure elucidation using the ozonolysis. And we can see that between, say, these PC3 cells, and here's a different cell line that's been inhibited, we can see the variation in the change in the fatty acid pool. So in terms of the technology, Philip's done a great job of trying to build this into a sort of an integrated workflow with a view to, could you do this routinely and develop a really deep dive into the fatty acid pool every time to discover these new fatty acids? How many fatty acids are actually there in a sample like this? So if you apply that workflow, which is kind of an automated version of the method that I described, and you splay that out, here's the difference that you can see in a volcano plot between LN cap cells, these cancer cells that have been, the control versus the SCD1 inhibited. And you can see this big difference that big change in the phenotype that sort of pops out. And what's interesting is that, these are the types of things that we were getting hints of before, a lot of change in these N12 species, and something that I won't have a lot of time to talk about today, also polyunsaturated species that are showing non-methylene interrupted patterns of unsaturation. So double bonds separated by more than one methylene. So it's easy to sort of conclude that, what we're looking at here is just something that's really aberrant in terms of cancer metabolism. So we thought we would then deploy this sort of deep dive into fatty acids to sort of things that people had studied more previously and perhaps closer to sort of healthy or regular or control metabolism. So we took some of the NIST reference plasma, and we do, the sort of some composition analysis as you would from a GC, but then we can now take each of these unsaturated some compositions of fatty acids and splay those out in isomer space. And what's really frightening is how many different double bond positions we can get evidence for. So just for the 18-1 family of isomers, up to 15 different double bond positions. We can sort of track their retention time behavior because, again, we can get that de novo identification. So we don't need standards to generate these retention time trends. And then we can use the information in the retention time to give us additional information. So, for example, cis or trans double bonds or evidence for chain branching in these unsaturates. And that enables you to build this horrendously complex picture of the diversity in the unsaturation and therefore just the diversity in the fatty acid pool that's present in, I guess, a previously very well studied reference material. And so to put that in some kind of context, we sort of tried to look at a range of studies that had been done in the past on reference plasma, but also non-reference plasma. And we think that the pool of fatty acids could be as much as, nearly 180 different fatty acids. And what's interesting here is that, many of these haven't been described before in plasma. And then there's a significant number that we can't find a record of, for example, in lipid maps or any of the other databases. But we have strong evidence based on the ozonolysis and the retention time behaviors that we know what these things are. So I guess just in the last part to say, if you think that you've got weird fatty acids, the place to look, according to the literature, is in vernix caseosa and skin lipids. So, this wonderful quote from Nicolaides' classic paper, 2 words characterize the uniqueness of skin lipids: complexity and perversity. And so I said at the beginning that, these N10 double bonds are sort of non-canonical. They're not in conventional metabolism, except in the sebaceous system of mammals and humans in particular. And so you can see that here. So, you know, there are some of these weird species that I've spoken about, say, for example, the fatty acids with double bonds in the 12 position. So just a quick summary of some work from a master's student, Prabhu, in the last couple of months. If you apply these same techniques to the fatty acids, you can identify upwards of 250 fatty acids present in vernix, just in the fatty acid pool itself. And you see a lot of these unusual fatty acid double bond positions. So what's interesting about this is, you know, the human sebaceous system, again, is making them. This is not a perversity of cancer or something else. This is in the repertoire of human lipid metabolism. And the last point is, you know, we still need to keep driving the technology, because not only do we need to now know the double bond positions, but we need to be able to identify the sites of chain branching, because it's another variation, another isomeric motif. So what you see here is we just add an iodine to our derivative. We fire the laser inside the mass spectrometer on the mass selected ions. We get a pattern that is based on radical directed dissociation and cleavage of the carbon-carbon bonds. And where we get a gap in those patterns, we can assign the position of chain branching. So what that leaves us with is, if we look at vernix caseosa, and I'm aligning here the data from both the ozonolysis experiment and the association experiment, two different injections on two different instruments, but using the same LC method and the same sample. We can identify, for example, in the saturated 17-0 pool, chain branching at the 4 position, as well as chain branching at the 15, the classical iso position. But what's more interesting is when we bring that into the unsaturated pool, we can now demonstrate unequivocally positions of double bonds at a particular retention time and assign the positions of chain branching. So in this case, at the retention time of 37.5 minutes, we have a double bond at the N11 position in a straight chain 17-0. But we can then, sorry, I skipped over that. But then when we switch to the 35-minute retention time, we see the gap, which allows us to identify the N11 and the anti-iso branch position. So we can now see these patterns where in unsaturated lipids, where odd chain lipids are sort of showing this anti-iso branching, whereas the even chain lipids are showing this iso branching, broadly speaking. And so to bring that full circle, that allows us, again, those two pieces of information about that molecular structure allows us to bring that back to the lipid metabolism, where we can see the desaturation of the odd chains, okay, favoring the anti-iso pathway, and the desaturation of the even chains following the iso pathway. So I hope I've been able to convince you that what we're seeing is a sort of an underestimation of the diversity of lipid structures, and that's almost entirely a mass spec problem, because many of these structures are isomeric. And so what we need to expand that toolbox. We need the right knife for the right job. And what I think is really key is to have techniques that allow that de novo structure elucidation. We've got to move beyond a reliance on aligning retention times with standards and collisional cross sections with standards and things like that, to have tools that can give us de novo information about molecular structure. And when we do that, everything we look at points to there's new discoveries to be made in biology. Why do we see this partition between iso and anti-iso chain branching associated with different double bond positions? And I guess I touched on this, the ability to use those lipid isomers and variation in those lipid isomers to be really tight markers for phenotypical change. And there's some really wonderful opportunities based on work like Perdita Barron's group using sebum in particular. And here's a little bit of my sebum just to close things out. So here's some of these really weird and wonderful fatty acids that I've taken off my own forehead, to use changes in these to identify different pathologies. But I guess what we're hoping to be able to do is use the specificity to actually link back to the underlying changes in metabolism that are associated with the pathology. So move just beyond this pattern recognition to a connection between the metabolic outcome. So with that, I'd like to finish up, and I'm really grateful to a wonderful research group that I get to work with, many of whom I acknowledged on the way through. Fantastic group of partners, both overseas and at home. Particularly wanted to acknowledge a wonderful partnership with Josef Vachka's group and Lucas Kuttleman in Prague on the sebum work, and work with Martin Sadowski on the cancer studies. We've had generous funding from the Australian Research Council and also in partnership with Waters. So with that, I'll conclude, and thank you for your time, and very happy to take any questions.
So thanks very much, Steve. That's a beautiful illustration of the use of derivatization and ion chemistry in biological mass spectrometry. It's I love a presentation which is full of mass spectra. So I very much enjoyed that. So the floor is open for questions. Maybe I don't see any questions there at the moment. So maybe I can start with the first question. So you know, a drawback of derivatization chemistry normally, whether it's inside the mass spec or outside, is side reactions, unexpected side reactions. So is that something you see with ozone derivatization?
Yeah, thanks, Bill. Yeah, I guess the chemistry we've seen inside the mass spectrometer has been very faithful to the classical ozonolysis mechanisms. Certainly there are variations in things like the rate of the reaction, where the abundance of those ions could be affected by the rest of the structure of the molecule. But generally speaking, what we like about the ozone is its predictability and its faithfulness to just tackling the carbon-carbon double bond. There are a few small variations on that chemistry. So again, there's two product ions. Do you see more of one or less of the other? Sometimes that's kind of useful. We can use that to sort of infer things about whether the double bond is cis and trans and things like that. In terms of the other derivatization chemistry, so the wet chemistry we've been doing with the AMPP, that's been very well trodden by others, and it's kind of one of the reasons that we like it. And in many of the ways, we like that derivatization because it's also simplifying. I think a number of the derivatization strategies that are out there at the moment, if you're doing them, before the mass spec, you're actually implicitly making a complex mixture more complex. What we like about, sort of simplifying to the fatty acid pool, if you like, is that, everything that's a fatty acid sort of picks up that same fixed charge, and everything behaves very well then chromatographically and mass spectrometrically.
Okay, maybe I could just have a quick follow on. What if you have a ketone group, do you get ketoenol tautomerism and then ozonolysis of the tautomer?
Yeah, I think it tends to be, so the short answer is I don't know, but the slightly longer answer is when, in the instances where we have seen double bonds sort of conjugated with the charge, it's deactivating. So we tend not to see then reactions at all under those circumstances. So if you have, a mobile proton, if you like, which is not that common in lipids, right? Because mostly the proton is sequestered somewhere on an oxygen or a nitrogen, but there are some cases where we've looked at that, then we tend not to see ozonolysis chemistry.
Okay, thanks, Steve. So we have a question now from Laura Mori. If you unmute yourself, you can ask your question directly to Steve. Yeah, ask your question, Laura. Okay, I can't hear Laura. So maybe I will ask the question.
Yeah.
What about ion mobility mass spectrometry for complex lipid composition? Do you use that with the ozonolysis?
Yeah, so there's a configuration that we have explored where we've done ozonolysis first. We've done that on the Agilent systems, where you can actually do ozonolysis almost as the ions are in the ion funnel before they enter the ion mobility. And so there you're sort of relying on the chromatography at the front end to do some of the cleanup and some of the separation. Then you're doing the ozonolysis reaction, and then you're using mobility to separate out some of the ozonolysis products. And that's effective in kind of a different way, in that, you know, one of the challenges with ion mobility of lipids is a lot of the variation I've been talking about is in a carbon-carbon chain, and the structure of those chains in a gaseous ion are not very well defined. You know, it's not like a native electrospray of an intact protein where there's a really well-defined three-dimensional structure and really, you know, well-defined collisional cross-section. These things are kind of flailing around like a bit of cooked spaghetti in the gas phase. So one of the advantages we see is then when you sort of cleave off one of those double bonds and you introduce the oxygen into that chain, you can then get some very strong charge dipole interactions within the molecule, and you actually lock down the structure. So, for example, those oxidation products coming from an N7 versus an N9 double bond, then, you know, not only have big differences in mass, but they also, you know, splay out in mobility space as well. So that's been effective. What we would really like to do, but we're on the path to, but we haven't achieved yet, is to do, you know, LC separation, mobility separation, and then ozonolysis. So a little bit of teaser data here. We've started work with the cyclic IMS, and we've been doing derivatization here with, and Bill will appreciate this, you know, classical picolinol esters, which we're not allowed to call picolinol esters anymore. But it turns out they're beautiful derivatives for mobility. And so you can see here three different fatty acids in, you know, two cis and two trans. And we can start in cyclic mobility, we can start to really splay those out in space. So these are preliminary data, but, you know, what looks really promising is the ability to, again, have some sort of complex lipid separation on the front end by chromatography, maybe mass selection, break the molecule down, look at the fragments by mobility, and then interrogate those fragments by ozonolysis. So if we could bring, or even photodissociation, bringing all those pieces together would be a super elegant solution in terms of, you know, really providing a top-down structure elucidation and leaving very few questions unanswered about how many isomers you have and what their structure is. So yes, very excited about ion mobility. I wouldn't want you to go away thinking that I was poo-pooing it in any way.
Okay, so maybe as well, maybe I'll ask a last question if there aren't any more. So you know, you showed us all those huge number of different double bond positions. Is it now, do you think we can now convince Avanti and Cayman to actually synthesize standards so we can be 100% sure that your probable identifications are definite identifications? Do you think that's a realistic goal?
I think up to a point, and thank you for reminding me, because I neglected to have Avanti on my acknowledgement slide, but I visited Avanti in 2019 and showed some of these preliminary data, particularly from the cancer work. And one of the things that they were able to do is to introduce more isomers into their phospholipid products. So, you know, you can now buy the N10 variant that I've shown you as a standard. So not just the unusual fatty acid, which is, you know, by itself is hard to get, but you can buy that installed in a glycerophospholipid. And I think that's really powerful, particularly for benchmarking techniques where you can't necessarily do the de novo structure elucidation. And I think the other thing that's been really powerful that Avanti have done is to really look at the SN isomers that are available as well, and really paying attention to the fact that there's a lot of biological variation in that isomeric dimension as well. And so they have a number of new products now that, you know, allow us access to those different types of variations. And that's helping us, I think, really tackle structural variation. So I think in answer to your question, Bill, it is possible. I think the key to it is to demonstrate that these molecules are relevant. They're not just a flight of fancy, that actually, you know, yours and my metabolism can make all of these things. And so, you know, a more standard assay needs to be alert to the fact that these things could be there, and that would require access to some reference material.
Yeah, I think that's
plus coming back to the metabolism, you know, what happens then if you take, you know, something like that N13 and feed that back into the cell? You know, glorious experiments would be to have, you know, unusual isomers with carbon-13 or something, and that would allow us to really not only understand, to convince ourselves they're there, but to actually convince ourselves where they're coming from and where they're going. What's the cell actually using them for?
Yeah, I think that those are two very important points. That as it were, the manufacturers have got to see the realization of the value of making a product to actually make it, and then, but once they have it, then there's a whole load of biochemistry to be explored. So I think we've more or less exhausted our time. So Steve, thanks once again on behalf of Lipid Maps and all the audience for a really lovely talk, and I really appreciated all the spectra. So thanks very much, Steve. And a final comment is the next seminar, Lipid Map seminar will be next month, and it'll be given by Professor Michael Gelb. Okay, thanks to everybody. Everybody have a good day and a good evening in Australia. See you, Steve. Bye-bye.
Bye.